Okay, let's continue with chapter 10 and protein synthesis, or also known as gene expression. Okay, now into translation. Translation of the message into mRNA into the amino acid language of proteins requires an interpreter. And that interpreter is the ribosome. That's what, well, I should say, that's what interpret, because the next sentence says the interpreter is type of RNA. It's that where the interpretation, interpretation takes place in the ribosome. And the transfer RNA assists in this translation. And here's another form of RNA called transfer RNA, or tRNA, to match amino acids and the appropriate codons to form a polypeptide. So these tRNA molecules have to perform two distinct functions: pick up the appropriate amino acid, recognize the appropriate codons in the mRNA, and the unique structure of tRNA molecules enables them to perform both tasks. So tRNA molecule made of RNA, about 800 nucleotides, twisted and folded upon itself. And it's partially single-stranded, partially double-stranded. On one end of the tRNA is a special triplet of bases called an anticodon. That's complementary to a codon triplet on mRNA. So if you had, so the first codon in the mRNA is AUG, the start codon. What would be the anticodon to that? UAC. AUG, UAC is in the anticodon, so it complementary fits the AUG. During translation, the anticodon in the tRNA recognizes a particular codon in the mRNA using the base pairing rules. At the other end of the tRNA is a site where one kind of amino acid is attached. Although all tRNA molecules are similar, there's slightly different versions of tRNA for each amino acid. So you have to have a tRNA for every single amino acid, and every codon that codes for amino acid has to have an anticodon on a tRNA that attaches to it. Like what was it? UCA is serine, so is UCC, so is UCG. So you have to have a tRNA for every single codon that is found in the genetic code. And how many codons code for amino acids? 61. So there are 61 different tRNAs, or 61 different tRNAs with different anticodons. And it could have the different anticodon, but the same amino acid, because remember the genetic code is redundant. All right. So there it is. Here's the anticodon down here. Three nucleotides, and here's the amino acid attachment site up here. And there's a simplified representation of it. Notice I said it's double-stranded in some places, single-stranded in other places. Okay. Shape of the tRNA molecule, it's an anticodon on one end and amino acid attachment at the other end, hints at how the molecule acts during translation. So what's major theme is the relationship of structure to function. Its particular structure determines its function. So ribosomes are the organelles in the cytoplasm that coordinate the function of mRNA and tRNA and actually make polypeptides. So a ribosome consists of two subunits. Each subunit is made up of proteins and a considerable amount of another type of RNA called ribosomal RNA, rRNA. So here was another type of RNA. We talked about a primary transcript, we talked about a messenger RNA, tRNA, ribosomal RNA, and actually ribosome, because that's a type of RNA also. So it's really like 5 different types of RNA we'll be talking about. Okay. A fully assembled ribosome is a binding site for mRNA on its small subunit, and a binding site for tRNA on its large subunit. So there you have the two subunits, and they come together on top of an mRNA. right? So here's the mRNA right here, and here is the tRNAs here that are bringing amino acids to the ribosome to be put in a linear array. So, same two phases as transcription: initiation, elongation, termination. Initiation brings together mRNA, the first amino acid, with its attached tRNA and two subunits of the ribosome. The mRNA has a cap and tail that helps the mRNA bind to the ribosome. Okay. So there's the cap, there's the start of the genetic message, here's the rest of the genetic message, here's the end. So AUG has to be up here, and one of the stop codons has to be down here. So an mRNA binds to a small ribosomal subunit, then a special initiator TNA binds to the start codon, where translation is to begin on the mRNA. Start codon AUG. So every mRNA starts with AUG. And the tRNA that binds to it is then UAC, and it carries methionine. A large ribosomal unit binds to a small one, creating a functional ribosome. Once initiation is complete, amino acids are added one by one to the first amino acid, and each addition, and this is an elongation process in the next figure right here. So you have methionine, the initiator tRNA, start codon, AUG, and then it brings methionine. So there's AUG right there, the start codon. There's the anticodon of the initiator tRNA, UAC, carries methionine, and then it moves over to the P site. And then there's another, and then it moves, and then the ribosome moves over, and the tRNA moves from the A site to the P site, leaves open that A site, and then another tRNA comes in that has the right anticodon for the next codon in the mRNA. Let's say it's UUU, because I know that's phenylalanine. So next codon is UUU. So here comes along an anticodon with AAA that carries phenylalanine. It goes to the A site. It binds there. A different enzyme inside the ribosome connects the phenylalanine to the methionine. Then another enzyme breaks the bond between the initiator TNA and methionine. It drifts away. The tRNA that had goes from alanine goes into the P site. So the ribosome moves along, and the tRNA for the phenylalanine goes into the P site, and then you've got another amino acid, another codon. I think it's that codes for another amino acid. Another tRNA comes along that has the correct anticodon that goes to the A site. They connect those two amino acids, break apart the previous one, and moves along. And step by step, elongation goes around, and the ribosome moves down the mRNA, reading every codon, and the tRNA coming along and bringing in the tRNA with the correct anticodon and attached to amino acid. Until what? Until when? Until it reaches a stop codon. That's a UAA, I think it's a stop codon. When a stop codon reaches the ribosome's A site, the complete polypeptide is freed, and the ribosome splits back into its subunits. And then we're all finished. So which of the following does not participate directly in translation? Ribosomes, transfer RNA, messenger RNA, or DNA? DNA doesn't participate directly in translation. It's been put away on a shelf back in the nucleus. It's back in the nucleus. It's got nothing to do with this. We copied the mRNA, we made the mRNA from one of the strands in DNA, but it has nothing to do with the actual translation process. Okay, the flow of genetic information itself from DNA to RNA protein. In eukaryotic cells, transcription occurs in the nucleus. RNA is processed in the nucleus before it enters the cytoplasm. Actually, I think RNA processing actually takes place in the nucleolus, that special little area inside the nucleus. Transcription and translation are a process whereby genes control the structure and activities of cells. Which organelle coordinates translation? Ribosomes. And there it is. Transcription, RNA processing, amino acid attachment. Well, this happens someplace else in the cytoplasm where the amino acid attaches to the correct tRNA. Initiation of translation, AUG, the first initiator tRNA attaches. Elongation, ribosome moves along and reads every codon in order, and the correct tRNA comes along to bring the correct amino acids. The amino acids are attached to each other in long row. Finally, we hit a stop codon. Then the polypeptide detaches from the ribosome breaks apart into two subunits, and actually the mRNA disintegrates. And then the protein goes ahead and does whatever its rules, and protein synthesis is over. So genes carry the instructions needed to build an RNA and then a protein. Which major theme is illustrated by this section? This is information flow, right? Genes to RNA to protein. Okay, now let's get to mutations. Any change in the nucleotide sequence of a cell's DNA is called mutation. And they can involve large region of chromosome or just a single nucleotide pair, as occurs in sickle cell disease. So, it can involve a lot of nucleotides or just one nucleotide. And so what I'm going to call structural mutations, because we're looking at mutations in two different ways. Structural mutations, and that's what the book talks about. And then it also talks about what effect a mutation will have on the organism itself. I'm calling that functional mutations. And there's three types of functional mutations. Neutral mutations that have no effect on the functioning of the organism. Deleterious mutations, which cause a serious problem in the functioning of the organism. Or beneficial mutations that actually help the functioning of the organism. And what they are mistakes in DNA replication. This doesn't happen in protein synthesis mutations. It happens in the S phase of the cell cycle when DNA is being copied. Because remember, DNA polymerase is not a perfect enzyme. It makes mistakes. Not very often, but it does make mistakes. And these structural type of mutations divide into two general categories: nucleotide substitutions and then nucleotide insertions or deletions. So let's look at this. Nucleotide substitutions involve one nucleotide. Insertions and deletions can involve one nucleotide or many nucleotides. Okay. So here's sickle cell disease. This is called a point mutation, and it's a substitution. So here's a normal hemoglobin DNA, CTT. Its complementary sequence in the mRNA is GAA. That codes for amino acid called glutamic acid. But then, in some individuals, and these just happen random, and they always happen, and they happen randomly. You can't predict them. That the DNA polymerase is going to make an error in copying DNA. Instead of having CTT here, it puts an A there instead. It made a mistake. It's a typo. Well, the corresponding codon in mRNA is GUA. What does that code for? Valine, not glutamic acid. It's a different amino acid. And in this case, it's a serious error because glutamic acid is a polar amino acid. Valine is a non-polar amino acid. It's quite a different type of amino acid. So it causes the sickle cell hemoglobin. It causes the hemoglobin to become sickle shaped in a low oxygen environment. So this is a missense mutation. It involves a single nucleotide and changes the amino acid coding. That's called missense. A nonsense mutation changes an amino acid codon into a stop codon. And what can that do? That can, so in this case, a point mutation can affect a lot of nucleotides because you can prematurely end a protein. A protein is supposed to be 100 amino acids long. If you have a nonsense mutation at like the 57th codon, then the amino acid is only going to be 56 amino acids long, because that 57th one was supposed to be like serine, but it converted into a stop codon. So now the protein is only 56 amino acids long when it should have been 100. So that's could be a much more serious situation. But missense just involves a single nucleotide and changes the amino acid coding. Now, sometimes you can have and it's called a substitution. So this is called a nucleotide substitution. And sometimes, though, you can have a nucleotide substitution that does not change the amino acid sequence of the protein. Because remember, the genetic code is redundant. Serine is coded for by 6 different codons. So you can change, it's usually the 3rd letter that can be changed, and it doesn't change amino acid. So you could have a substitution in one codon and not change amino acid that is put into the protein. So that is as far as functional mutation goes, that is a neutral mutation and will have no effect on the organism whatsoever, because it's exactly the same protein. After mutation, you're making exactly the same protein with exactly the same sequence, even though there was a change in the DNA. And this can be used. Remember, we talked about making those DNA, evolutionary trees, those relationship trees. That's what they use to make these neutral mutations that change the DNA, but don't change the protein that's coded for that DNA. And like I said, they happen all the time. Okay. Mutations involving the deletion or insertion of one or more nucleotides in a gene, these are called frameshift mutations. And they often have disastrous effects. I mean, huge effects. Because it changes what's called a reading frame. So usually, you insert a nucleotide, you insert a letter, or you delete a letter. And that changes that codon, and every codon was called downstream from that. So you can think of upstream and downstream. Upstream is towards you're in the middle of the message. Upstream is toward the start codon. Downstream is toward the stop codon. So if you have an insertion and deletion here, then every nucleotide downstream from it is changed. Well, if you have a mRNA coding for amino acid that's 100 amino acids long, and it happens in the 99th, and they have an insertion in the 99th codon, then they're going to change that codon and the 100th codon, right? But if it happens in the 50th codon, then you're going to change that and all 51 past that. All the 50 codons past that. So it can have a really big effect on the, so here they are. Here's a normal amino acid. Here is a base substitution. You changed a G to an A. Right? Here's a nucleotide, so that changes that, just changes that one. So this is a missense. It just changes one nucleotide, which change one amino acid in the protein. So you have a different amino acid. You should have a glycine, now you have a serine. But that might change too much because serine is a polar amino acid, so is glycine. And here's the thing also, sometimes you can actually change, many times you can change amino acid sequence of protein and it doesn't change the functioning of protein because it doesn't happen in the active site. Say it's an enzyme and you change the shape of a little part of another part of the enzyme is changes shape. But the active site stays the same shape. Then even though you change the amino acid sequence of the protein, you still didn't change the function. So that's a neutral mutation also, even though you change the amino acid sequence. But sometimes even a change of one amino acid can lead to a big change in the function of the organism, like sickle cell. One, hemoglobin is 455 amino acids. Sickle cell changes one amino acid, and it has this big effect. So, it's not predictable. You can't tell whether this change in this one amino acid is going to affect the organism or not. This is part of what's called natural selection. You get the organism will go out in the environment and see whether it survives or not. If it doesn't survive, looks like it was a bad change, deleterious effect. If it survives, maybe it's neutral. If it does better than it did before, it has more offspring than it did before, it's healthier than it was before, maybe it was a beneficial mutation. You can never tell. It has to be selected for in the environment. Okay, here's the insertion. As we can see, or deletion. We took out a U. So that changed that. It used to be UUU, now it's UUG. The one past that was GGC. Now it's GCG. So it changed, like I said, every codon downstream from where that deletion occurred. Insertion is the same thing. You inserted it. It was UUU. Now you inserted a G. Now it's UUG. And the second codon down from that used to be GGC. Now it's UGG. And so on and so forth downstream. So that's why insertions and deletions, these so-called frame shift mutations, can have huge effects on making a really different protein than you had intended to. And it's usually pretty deleterious. That big of an effect is usually deleterious. It's a bad mutation. Okay. Mutations can occur in a number of ways. Spontaneous mutations result from random errors because, DNA polymerase is not a perfect enzyme. But other times you can actually increase the rate of mutation by being exposed to certain physical and chemical agents called mutagens. The most common is radiation. Radiation can cause mutations, can increase the rate of mutations. Chemical mutations. Many mutagens, these are chemicals that can cause mutations. And some of these mutations can cause cancer. So some mutagens also are known as carcinogens, agents that cause cancer. So you need to avoid them as much as possible. Like asbestos is a carcinogen. Okay, although mutations are often harmful, like I said, the most common mutations are neutral. The second most common are harmful. But the rarest are beneficial, and they can actually help the organism or help the organism do something brand new, add novelty. And so mutations are the one source of the rich diversity of genes in the living world, a diversity that makes evolution by natural selection possible. Where do new alleles come from? Well, mistakes in DNA replication. That's where they come from. Alleles come from mutations, right? So everybody had brown eyes one time, you had a mutation, now people have blue eyes. So that's where new alleles come from. So this is, like I said, it's the raw working material of evolution. And it's really one of the few things in evolution that's actually random. It's completely random. Most people think that evolution is completely random. No, it's not. Natural selection is not random at all. It's called selection. The random part. And there is mutations. There's also ways populations can change randomly. But natural selection is the most important type of evolution, and it's not random at all. So these mutations happen to organisms. The organisms go out in the natural world and see what happens to them. Do they survive or not? So, it's a good thing. DNA polymerase is not a perfect enzyme, because if it evolved first and it was perfect and it never made a mistake, we wouldn't be here talking to each other. Because the only kind of life on Earth would be these primitive cells that have perfect enzymes that never make a mistake. And that would be it. There wouldn't be any evolution of life on Earth. So checkpoint. What would happen if a mutation changed the start codon to some other codon? Well, then that would be a big problem, because then the ribosome wouldn't know where to begin. And that whole protein wouldn't be able to be translated, I don't think, because the ribosome wouldn't know where to start. So that would be a big, serious mutation. What happens if one nucleotide is lost in the middle of a gene? That would be a nucleotide deletion. That would mean that codon is changed, plus every codon, and therefore every amino acid downstream from that mutation has been changed. Okay, mutations and diversity. Okay, now we're going to get into viruses. Talk about viruses. Infectious particle consisting of little more than genes in a box, a bit of nucleic acid wrapped in a protein coat. And sometimes it's an envelope of membrane. It almost looks like a cell, but it's not. They share some of the characteristics of living things as having genetic material, but it's not considered alive because it's not cellular and it can't reproduce on its own. Viral genome and actually the viral genome is more varied than living things, because a viral genome could be single-stranded DNA, double-stranded DNA, single RNA, or double-stranded RNA. So most, like this cold virus here, is the adenovirus, or also a rhinovirus. It's just a DNA with a protein code. That's it. Really simple things. Now, viruses attack bacteria called bacteriophages, or phage for short. Consists of a molecule of DNA enclosed with an elaborate structure of proteins. And once they infect the bacterium, most phages enter a reproductive cycle called the lytic cycle. That is, they hijack the nucleic acid and protein synthesizing machinery of the bacterium. They make new viral particles. They break open a bacterium, killing it, and then these particles go on to infect other bacteria. But some viruses can also reproduce by an alternative route called a lysogenic cycle, in which viral DNA replication occurs without the death of the cell. You can make, and these viruses get incorporated into the chromosome of the cell that it invades and becomes part of that organism's genome, and is passed on to its offspring. And sometimes they enter a lytic cycle and they kill the cell, but other times they don't. They can actually make new viral particles and excitose those viral particles and not kill the cell they're in. And those are the kind of viruses that if you get one, you get it for the rest of your life. Lytic viruses, like a cold virus, it's lytic, it kills some cells in your nasal passages, but you can replace those cells. Eventually, the immune system can attack these viruses and completely eliminate it from your body, right? The lysogenic viruses, once they get inside your cells, they stay there. They incorporate their genes into your chromosomes and you have them for life. That's it. Herpes viruses, good example. Epstein-Barr virus, good example. HIV is also lysogenic. You get it for life, right? But a lot of these viruses are not deadly. The ones that have been lysogenic, because we've evolved with them. So a herpes virus might be embarrassing or and cause blisters on your skin, but it doesn't kill you. Epstein-Barr also will not kill you. Epstein-Barr can cause mottling, you can get pretty sick, but it's not going to kill you. HIV, however, emergent virus, will kill you if you don't get drugs against it. But if it stays with us long enough, it may become one that's also not dangerous. But right now, it is lethal if it's not treated or not, with the drug treatment. So bacteriophages, they look like little spacecraft, and they inject their DNA into the bacteria. So here's the thing. Our bacteriophages, so they attack bacteria. We also have plant viruses that attack plants. Do you have anything to worry about if you come into contact with a bacteriophage? No, you don't. Because it attaches to bacterial proteins on the outside surface of the bacteria. And it's specific, it's like an enzyme. It fits into that. We don't have those proteins on our cells. So bacteriophages cannot attack to our cells. So as a matter of fact, viruses are usually pretty specific to what they attack. They attack animal viruses attack animals, plant viruses attack plants, bacteriophages attack bacteria. And sometimes also viruses can only attach to one type of cell in this animal, like a hepatitis virus. It only attaches to liver cells. HIV only attaches to what's called a helper T cell. That's it. Polioviruses only attach to motor neurons and so on. So herpes viruses actually attack the neurons. That's it. And it's a good thing too, because every time you go swimming in the ocean, you're swimming in a sea of phage viruses. I mean, there's trillions of them in every drop of seawater. It was discovered by this woman scientist who wondered why there wasn't a lot of bacteria in seawater. Because there's all kinds of nutrients. It should be covered with bacteria. It's because it's a sea of bacteriophages that are killing bacteria. So you don't have to worry about phage viruses. Okay. So here's two types of cycle for a phage named lambda that can infect E. coli bacteria. Escherichia coli, it's a coliform, it's a fecal bacteria. We're going to describe one way that some viruses can perpetuate their genes without immediately destroying the cells they infect. They, like I said, incorporate their genes into our chromosomes, and they can sit there for years and years and not do anything, and matter of fact, never ever do it. A lot of us are infected by Epstein-Barr and will never get mono, but they're there and they never become active. Infected by a herpes virus, it gives you cold sores. Every once in a while, it pops out, it makes blisters, it makes active virus, then goes by, and then doesn't come back. And it might not come back for years and years, or maybe it never come back. Fortunately, like I said, herpes viruses, they don't kill the cells that they're in. So they're exocytosed from these nerve cells, as opposed to polioviruses, which are lytic. We don't have to worry about polio anymore, but when I was a kid, we really had to worry about it because it infected motor neurons, and it was a lytic virus. So when it infected motor neurons, it destroyed, and when it made new particles, it burst the cell, it destroyed that motor neuron. And once you lose a motor neuron, you don't get it back. So it would paralyze. People were paralyzed if they got infected with poliovirus. Fortunately, we had a vaccine, and we pretty much eliminated polio from the United States, which is really a good thing. Okay, so there's the two cycles. Here's a lytic cycle, attaches to cell, injects DNA, new viral particles are made. The phage is the same. Cell lysis, which breaks apart, sends particles to new places and infects new cells. Or it incorporates the phage DNA is encoded in the bacterial chromosome. It's replicated with each replication of bacteria. The phage DNA these genes are passed along, and just stays in the bacteria forever, and it may never do anything. But eventually it might enter a phase cycle and eventually kill the cell. So viruses infect plant cells, can stunt plant growth and diminish crop yields. They usually have RNA rather than DNA as your genetic material. Like the tobacco mosaic virus. Actually, I think that was like the first virus that was ever discovered. There's no cure for most viral plant diseases. There's no cure for most viral diseases. If we have antibiotics, well, we have a lot of antiviral drugs that can keep it under control. Like HIV infection is now a treatable disease, and you can live a fairly normal life by taking the right drugs. But you can't cure it, particularly lysogenic viruses. Like I said, you have them for life. You can't get rid of them. So agricultural scientists focus on preventing infection and breeding by genetically engineering varieties of crop plants that resist a viral infection. There's the tobacco mosaic virus. So viruses infect animal cells, common cause of disease. No virus greater human threat than the influenza virus. Well, now we have the coronavirus. Animal virus is outer membrane and fossil membrane, envelope and labels the virus to enter and leave a host cell. Actually, viruses trick our cells. They bind to certain receptors that cause our cells to endocytose the virus into the cell. So many viruses, including what's called the flu, common cold, measles, mumps, AIDS, polio, have RNA as a genetic material. Diseases caused by DNA viruses include hepatitis, chickenpox, and herpes infections. There's the influenza virus. It has a membranous envelope plus the coprotein and the RNA. Here's the reproductive cycle of an enveloped virus, a mumps virus, entry, uncoding, virus. Okay, here's the thing. So viruses that RNA for the genetic material have a problem when you infect our cells because they have to reproduce RNA. Well, we make RNA from DNA. We don't make RNA from RNA, or we don't make DNA from RNA. So how are they going to hijack? So they can't hijack our material to make their RNA, to make RNA from their RNA. So what they need to do is they bring an enzyme with them called reverse transcriptase that can, either it's an RNA synthesis that can make, that's for HIV. For an RNA enzyme, they bring an enzyme with them that can make RNA from RNA. And HIV brings an enzyme called reverse transcriptase that makes DNA from RNA. And then, so that DNA is made from the viral RNA. Then they can use our RNA polymerase to make their RNA from their DNA, and then make new viral particles. So that's a problem. RNA enzymes have to bring along inside, so sometimes inside viral particles, there's an enzyme in there too, a viral enzyme that they need to make RNA from RNA or make DNA from RNA. And here they can exocytose. So the mumps virus, it has a lytic cycle, but it makes new viral particles, but it doesn't burst the cell that's in. It doesn't destroy the cell that's in. And it's not a lysogenic virus. It doesn't stay with you. It doesn't incorporate its genes into your chromosomes. It just makes new viral particles and infects other cells. And eventually you can get rid of these viruses that do things like that, like flu viruses, like cold viruses. They're always outside cells. They don't stay inside cells. So they can be destroyed because the immune system can't destroy viruses that get inside cells and stay there. That's why I got them for life. They can only kill, well, deactivate, destroy viral particles that are outside of cells. And so that's why you can completely get rid of coronavirus, completely get rid of flu viruses, cold virus, completely get rid of it, because they don't stay inside cells, they go outside cells, and eventually your immune system can wipe them out, can just take out every single one. But not the lysogenic viruses. They stay within cells, and so therefore your immune system can't get rid of them. So herpes viruses called chickenpox, shingles, cold sore, genital herpes are enveloped DNA viruses that reproduce in a host cell's nucleus, get to envelopes in the cell's nuclear membrane, and copies of the herpes virus DNA remain behind in the nucleus in your own chromosomes, where they can remain latent for long periods of time and may never become active and make blisters again, or maybe every once in a while they make blisters. But like I said, they're not lethal. So they're not really dangerous viruses. They may be a little embarrassing or something, but they're not dangerous. The amount of damage a virus causes depends upon how quickly the immune system responds to fight the infection and the ability of the infected tissue to repair itself. We recover completely from colds because our respiratory tract tissue can effectively replace the damaged cells. So you lose these cells in your respiratory tract. They're replaceable, okay? But as I mentioned before, the polio virus attacks nerve cells, and it's lytic, and it destroys the nerve cells. So those damage is permanent. And the only medical option to prevent this disease is with vaccine. So I got the polio vaccine. We all did, and now we don't have to. And now people don't have to be vaccinated by polio anymore because we completely wiped it out in the U.S. So same thing with smallpox. We used to have the smallpox. That was a scourge. That was completely wiped out by vaccinations. So West Nile virus. So we have these new viruses always popping up, and new variants of virus. You know, we have the variants of coronavirus. These are mutations. Their DNA polymerase or RNA polymerase also makes mistakes, and so that's where these new variants come from, mutations. And these mutations from the virus's standpoint might be beneficial because it makes it more infectious and more likely to be passed on. You know, like the South African strain of the coronavirus, it's a little easier to catch it. That would be a beneficial, it's deleterious for us, beneficial for them, because the whole point of the virus is to pass itself on, get into other individuals and make new viruses. That's the only thing the virus wants. Some viruses spread by mosquitoes. There are vectors, wild vectors that have like the rabies virus. It's out there in wild animals. There are certain they're called vectors. They can carry the virus, but they're not made sick by it. All right, and so they can pass it on. We are vectors for Epstein-Barr because it's made in salivary glands, and if you share some spit with somebody that's infected with Epstein-Barr, congratulations, you're affected by Epstein-Barr. But it doesn't cause disease, and most of it doesn't cause disease. 85% of us are infected with this virus, and it almost never causes disease. Every once in a while, somebody comes down with mono, and you're really sick, you know, and you're miserable for a while, and you're weak, but it goes away. You recover from it. And for some viral, I don't know too much about this because I don't know much about virus, but some viruses are really tough to make a vaccine for. And other ones, it's easy. And so particularly for West Nile, apparently it was really tough to make a vaccine. Okay, so I'm not going to go through this this procedure where they tested a vaccine for West Nile. But I did want to talk about AIDS, acquired immunodeficiency syndrome, HIV virus. It's a retrovirus. An RNA virus that reproduces by means of a DNA molecule. They reverse the usual flow. They make DNA from RNA with that reverse transcriptase. Well, there it is in the next bullet, reverse transcriptase. They gotta carry that along, because remember, we don't make DNA from RNA. We make RNA from DNA. We make DNA from DNA. So they have to carry this and first they make a viral DNA from their RNA, and then they can hijack our material to make their viral particles using our RNA polymerase. And so here's the HIV virus. It has a surface proteins and a phospholipid envelope also. And there it is, viral RNA, and there's the reverse transcriptase, makes a DNA strand, double-stranded DNA, puts together viral particles. It gets incorporated into the chromosome of things called dendritic cells and helper T cells. And it shows it being exocytosed. No, it kills T cells. And that's what causes AIDS, is the destruction of these T helper T cells. They're the most important cell in the immune system. They help all other parts of the immune system, hence the term helper T. They're the most crucial cell in the immune system. And eventually, and it takes like 7-8 years for you to develop AIDS, because you have a certain level of T cells, and it gets dropped because of the HIV destroys these T cells. You make new ones, and it drops it again to make new ones. But after years and years, your level of T cells drop and drops and drops. After about 7 or 8 years, it drops so low, you cannot protect yourselves against any kind of disease, infectious disease. And people start getting these diseases that like nobody gets, like Kaposi's sarcoma or something, that nobody gets unless your immune system has been destroyed. And so HIV doesn't kill you. What kills you is the opportunistic infections you come down with because your immune system has been destroyed. But now, so as I mentioned, it kills several types of white blood cells important, particularly T helper cells. No cure for AIDS, but now they have antiviral drugs. One of the drugs inhibits reverse transcriptase. One and there it is there, the second type. The first type inhibits an enzyme called proteases. HIV is strange. It copies its RNA from the DNA it made from its RNA for reverse transcriptase, and it makes this huge mRNA molecule that is like 3 proteins in one, codes for 3 proteins in one. We don't do that. Every mRNA codes for one particular protein. You can make several different mRNAs from a gene, but each mRNA just codes for one protein. But HIV makes a huge mRNA that codes for 3 proteins. So then you have to have these viral enzymes that will cleave this huge mRNA into 3 separate mRNAs that code for 3 separate, that go to ribosomes, and you make 3 separate viral proteins from that. But now they have a drug that will inhibit that protease. And therefore, you can't break that big mRNA into 3 separate individual mRNAs. And therefore, that huge mRNA cannot be transcribed at the ribosome, and that stops the viral proliferation. And now these drugs, and there's a third drug also that's an artificial like guanine. So now combinations of these 3 drugs have turned HIV infection into a manageable illness, and that you can live, and you can drop your viral load to practically nothing. As long as you keep on taking these drugs at very precise intervals every day for the rest of your life. But so now it's a controlled illness, and this is all due to, and they use 3 rather than just 1, because if you keep on using 1 drug, you can get resistance. It's just like penicillin. All bacteria are resistant to it. So you could get, if you just use 1 drug, the virus could eventually develop a resistance to it, but use 3 drugs, each one doing a different thing. And it's very unlikely that this virus is going to evolve a way to get past 3 different types of drugs at the same time, become resistant to 3 different, very different drugs at the same time. So this is why it works. So, okay, so why is HIV called a retrovirus? Because it has RNA for its genetic material, and it has to make a DNA from RNA, which is retro, which is reverse the normal direction. Okay, so as I said, it's much more effective than individual drugs because it's very unlikely, like I said, a virus can develop resistance to 3 different drugs at the same time. So, but it still doesn't get rid of the drug in the body, of the virus in the body. It's still there, it's just not active. And you can test these people's blood, and you can't find any virus in their blood because it's not outside cells, because you haven't made new viral particles, but it's still in there. So it has no cure yet. Prevention, avoidance of unprotected sex with an infected person, and needle sharing is the way to completely avoid, because a lot of people got HIV from the drug addicts that's sharing needles without if you come into contact with the body fluid of a person that has HIV infection, and you're likely to get it. Okay, the last thing, prions, bizarro things. Infectious protein that cause brain diseases in several animal species. When they first discovered it, they couldn't believe it because they thought we needed something with nucleic acid to be infectious. These are proteins from the nerve tissue of an infected animal that gets into to the brain and persuades other, and they're misshapen, and they persuade other brain proteins to become misshapen, and it destroys the animal's brain. Converts normal proteins into misfolded versions. Misfolded proteins come together, disrupting brain functions, and the only hope lies in understanding the process of infection and not to eat infected material. And every mammal has its own type of what are called encephalopathies, like bovine encephalopathy is mad cow disease. You got mad cow disease. Sheep has something called scapie. Deer has something called wasting disease. So if you go out and kill a deer, you got to bring it into a station and have it tested before you eat the meat. And people have a variation called Creutzfeldt-Jakob disease. And if you eat infected nerve tissue from an infected animal, these prions will go to your brain, misshape the proteins in your brain, and destroy your brain and kill you. And there's nothing you can do about it if you get infected with these things. Okay. Finally, we already talked about emerging viruses, Zika, West Nile, because viruses mutate. And new virus diseases or viruses can spreading of existing viruses to other ones. Viral diseases from a small isolated population can spread, leading to an epidemic or a pandemic, which is what we have now. Coronavirus, emerging virus. Apparently come out of Wuhan, China, and spread across the world, and we've got this. And so this is not going to be, we're going to deal with this, take care of it. It's not going to be the last one. We new ones all the time. So we just have to deal with them as they come along, the best we can. So there's some major emerging viruses. AIDS virus, 1981. H1N1, West Nile, severe acute respiratory syndrome, avian flu, Ebola, Zika. And now we add coronavirus to that list. Right? So we live in an illusionary combination with virus. There's no chance that we survive the fight against emerging viruses. Understanding requires understanding molecular biology and evolutionary processes to make vaccines that can work to defeat these guys. And that's what we're doing finally with coronavirus. And that's it, folks. So this is two. This was kind of a long one. So this is going to be 106 DNA 2, and this is going to be, like I said, information for the extra credit exam. If you want to take the extra credit exam, remember, it's not mandatory. It's all up to you whether you want to take it or not. Okay. I'm going to cut it off here, and we'll see you later.